Good morning, everyone. It's a pleasure to give this talk today. So I am going to be talking about unraveling the genomic complexity of common and rare genetic disorders. And I'm going to be discussing some of our approaches to how we try to discover causal variants in a few different types of diseases. So I'm going to start this presentation off with a quote from probably one of the most prolific geneticists these days. And it's from Marie-Claire King. And the quote says, "Exponential population growth fueled by the development of agriculture in the past 10,000 years and of urbanization in the past 700 years has resulted in a vast number of new alleles. Collectively, these alleles have generated an immense degree of genetic variation. And given the size of the present-day human population, every point mutation compatible with life is likely present in someone, somewhere." So I really like this quote because I think it motivates a lot of what we'll be discussing today. And if we think about this for a second, what it actually implies is that there may be a whole host of very individually private rare traits that are caused by some of these very rare mutations that are occurring in someone somewhere in our population today, and that kind of motivates how we go about looking at the genetics of rare disease. So in the talk today, I'm going to first describe two of the two paradigms that exist today to explain the genetics of complex human disease. I'll be discussing how multiple strategies were used in one of our studies to uncover the genetics of a complex disease and the importance of using multiple strategies. For that, I would use the example of hereditary hearing loss. I'll follow by discussing some of the unique challenges of rare diseases and discovering the genetics behind rare diseases, and I'll use another example of a disease called NGLY1 deficiency. And then finally, I'm going to tell you about some of our work at the Rare Genomics Institute, where we're starting to build a research model to overcome some of the challenges that we've had to deal with in trying to discover the genetics of rare disease. So two paradigms have emerged to explain the genetics of complex human disease. The first one is probably the most obvious and intuitive, and that is simply that we can assume that everyone who shares a disease actually shares the same underlying genetic cause. So that could mean that everyone who has that disease has the exact same mutation, or mutations in the same gene, or even mutations in the same set of genes that might form a biological pathway. And there are many, many examples of this in the literature. Probably the most well-known textbook example of this is the sickle cell anemia trait, in which everyone who has that disease actually has the exact same mutation. And then the second paradigm, which is probably less intuitive and less obvious, and only in the last few years have we really started to see this play out in terms of discoveries, is that everyone who has a disease that on the systemic or physiological level appears to be the same disease actually has their own private underlying genetic cause. And when we think about what's happening in real life, or in real life situations of complex disease, we imagine that we have some sort of balance between these two paradigms. So that is, in a population suffering from some disease, you might have some subset of that population all sharing some common genetic cause, whereas other members of that population might actually have their own private genetic cause. And the second paradigm I thought was very well, very beautifully illustrated by another quote from Marie-Claire King in that same paper, where she uses the analogy of Tolstoy, and she says, "Every unhappy family is unhappy in its own way." And this is from Tolstoy, and he was obviously reflecting on the unique nature of human tragedy in that statement. But Marie-Claire King used this as an example to demonstrate sort of the misfortune of human disease. And she says, from the perspective of genetics, we suggest that complex human disease is in fact a collection of individually rare, even private conditions. And that's at the genetic level, whereas at the systemic level, the overall symptoms may actually look very much the same. So given that we have an understanding now of these two paradigms, I'm going to talk about hearing loss. And the process of hearing is obviously very involved. I mean, we have this very intricate structure, the ear, which takes mechanical signals from sound waves and converts those to electrical and chemical signals in neurons, and that's what actually allows us to hear. So if we think about what goes wrong when we don't hear, so in the case of hearing loss, we find that this could actually be clinically very complex. So hearing loss manifests in a number of different ways. It can be syndromic or non-syndromic, meaning that it can occur in the presence of other symptoms or simply by itself. The severity can range from mild to profound. The part of the ear that's actually affected can define the subtype of hearing loss. It can affect one or both ears. It can be inherited in a number of different patterns, and it can occur at at birth or develop over time. And obviously, this is just a demonstration of how all of the different factors that can go into describing this phenotype. But at the end of the day, the outcome that we measure is whether or not someone is hearing and at what frequencies they are hearing or not hearing. And for the purposes of this talk, the type of hearing loss that we decided to study is sensorineural congenital hearing loss. So these are patients who have all developed hearing loss either at birth or before they began to speak. So hereditary hearing loss is actually one of the most prevalent human sensory disorders. About the majority of cases actually are non-syndromic, so that means that the only symptom they show is hearing loss. And so far, most of the most hearing loss cases have been found to be caused by a single gene. One of the really interesting aspects about the genetics of hearing loss is the fact that so far there have been 133 candidate loci encompassing 78 genes. I have that number there, but that number keeps growing. And that's actually a very, very large number of genes to be influencing one trait. And what's even more interesting is that about half of the severe to profound cases of autosomal recessive non-syndromic hearing loss are actually due to mutations in one single gene, and that gene is GJB2. But the other half of the cases are due to mutations in a huge array of different genes. And so this led us to our first hypothesis, which is basically that there may be many other yet to be discovered genetic contributors that underlie a significant percentage of these cases. And we set out to study a cohort of samples for which they had all been tested by standard hearing panels, and they were all found to not have mutations in GJB2 or any of the other common hearing loss genes, and all of these cases have remained elusive until now. The other thing I'd like to say about hearing loss genetics is that the methods that people have used to discover these genes so far have generally been small studies within families or studies of linkage analysis within families. There haven't really been too many very large association type studies with hearing loss, and most of the mutations that have been discovered or have been looked for were single nucleotide variants, and single mutations in single genes. So as we know, human genetic variation is vast, and there are many types of variation besides just single nucleotide variations, which is shown up here. We also have short indels, and of course there's this whole world of structural variation, including copy number variations and inversions and translocations. And so far, when we did this work, no one had systematically looked for the involvement of any of these structural variations in herring. And there had only been one study published that demonstrated a deletion in a known hearing loss gene as a cause of hearing loss. So that led to our second hypothesis. We had done a lot of work in mapping copy number variation in the human genome. And from this work, we found that copy number variations are actually enriched in genes that are involved in sensory perception of the environment. And this includes things like taste and smell receptors. And so it made sense to us that CNVs may be involved in hearing. So we also set out to search for CNV involvement in the genetics of hearing loss. And so I told you that we took a multi-strategy approach. So this is basically what we did. We used two different variant mapping technologies. We used exome sequencing, and we also used very high resolution array comparative genome hybridization, which was for copy number calling. And because of these two paradigms that I described previously, we decided to do both familial and association strategies. So we looked at a large cohort of cases and controls, and we also looked at individual families suffering from different types of hearing loss and followed the genetics through those. And in total, we did this on 3 families that had different severities of hearing loss, and we had 150 probands as well as the same number of controls. So here are some of the families that we used. So family 1 was a family that showed classic recessive inheritance of hearing loss. This family suffered from very severe hearing loss with a second phenotype of megalocornea, which means an enlarged cornea. Family 2 suffered from moderate hearing loss, and there was actually a son in this family who had various other congenital abnormalities, and the hearing loss in this son was actually a different type than what was in these twins, and so we ended up ignoring this child in this analysis. And then family 3 had an interesting mild situation of hearing loss where we didn't have exome sequence data for the grandparents, but we did for the rest of the family. And the mother was affected, but not as severely as the children. So it seemed to be increasing over the generations. And finally, we had a bunch of 15 individual probands, which we didn't have any familial information about, but we performed exome sequencing on all of these anyway to try to figure out what we can find. And so this was our general strategy. We started with, we used an Agilent capture kit for the exome data. We did paired-end sequencing on a HiSeq. And from that data, we did 2 different alignment and variant calling processes. So we used DNA nexus to do read alignment and SNP and indel calling, and we also used real-time genomics on the family, which used essentially a very different process than DNA nexus in order to do variant calling and de novo variant calling as well. And then we combined those variants that we found. We used ingenuity variant analysis to filter the variants, and then we found our candidate variants from that. And on the right, I'm showing the general filtering scheme that we use for the variants. So in each situation, we had thousands and thousands of variants. We applied quality filters to the data. We applied frequency filters to the data. So this is we removed variants that were very common in normal populations because we assumed that these cases of hearing loss were so rare that that you wouldn't find these alleles at high frequency in normal populations. We used a filter to predict deleteriousness, and I'll describe this more as we go on. We also filtered based on the genetic inheritance pattern of the of what we expected based on the family structure. We used biological filters based on the phenotype, which was hearing loss, and this led us to various candidate variants. So I'm going to tell you quickly about these three families and what we did. So for family one, this was probably our most successful case. We started with all the variants. We went through this filtering scheme, and this is a clear recessive mode of inheritance. And at the end of the day, we came up with one potential candidate gene, which was a compound which housed compound heterozygous missense mutations. And this gene was MYH7B. So MYH7B is actually a myosin heavy chain protein. And the two alleles that we found, so the paternal allele, so myosin 7 has two functional domains, this head domain shown in blue and this tail domain shown in green. And it turned out that our two missense mutations actually fell within these functional domains, making it more likely that these were actually going to cause some kind of phenotype. So the paternal allele fell in the tail domain, and the maternal allele fell in this relay loop of the main head domain. And the table shows some very interesting properties of these variants. First of all, if we look in the 1,000 genomes frequency, 1,000 genomes populations, and the exome sequencing project from NHLBI population, we see that one of these variants, the paternal allele, was just not seen at all. So this appears to be private to this family. And the maternal allele was seen at a very, very low frequency. Both of these mutations actually showed very high scores in all the algorithms that predict how deleterious these alleles would be. And on the bottom, I'm just showing the Sanger validation tracks, showing that in this patient, F13, who's actually heterozygous for both of these alleles. So this one we thought was pretty clear, and then we did some more research into this gene that provided some corroborating evidence for this gene in hearing. So first we looked at the Allen Brain Atlas, and we saw that this indicated high expression of this gene in the primary auditory cortex and other regions of the auditory pathways in adult humans. Then we saw that this gene was actually concordantly expressed in mouse embryonic inner ear tissue in a similar pattern as another gene that's required for hair cell differentiation, but it wasn't expressed in non-ear tissue. And this was in a mouse. And then finally, we saw that in cultured rat hippocampal neurons, we saw a reduction in MYH7B expression. And we saw that this actually caused profound alterations to the dendritic spine morphology and excitatory synaptic strength in the actin cytoskeleton, indicating that potentially mutations in this gene or knockdowns of these genes would actually have some neurological effects in a patient. So we thought this was pretty solid evidence, given that not much else was known about this gene, and so that led us to predict this model. Which so we know what we do know about this gene is that it forms dimers, or the protein forms dimers, and so we can imagine that if the father has a mutation, a heterozygous mutation in the tail domain, and the mother has a heterozygous mutation in the head domain, the father is still able to form dimers of this protein where you can have two normal copies, and same with the mother. But the children are unable to form any normal dimers. So either they have two two mutated head domains, a mutated head and a mutated tail, or two mutated tail domains. And so we can see how in this compound heterozygous situation, maybe not having any ability to form normal dimers is what might be driving the phenotype. So this was probably the most fleshed out situation that we had. The second situation was a family with moderate hearing loss. And in this one, we performed the filtering the same way. We actually combined the results of DNA nexus and real-time genomics and used the union set of all variants that were called in order to do our filtering. That turned out to not provide any good candidates for this family, and so we actually ended up using only the DNA nexus variants, and that was the only situation in which we found two potential candidate genes. These were dominant heterozygous de novo mutations of two genes for which not much was known. So this kind of led us to, well, these two genes might be potential causative candidates, but we can't really say that much more about these two genes right now. And then in family 3, we were probably even, I would say, less able to nail it down, in that we found by a common filtering scheme. Of course, in this case, we looked for dominant mutations, and we found 6 potential candidates. These were all heterozygous missense mutations in genes that were located in networks containing a known hearing loss gene. So this was we looked at all the known hearing loss genes. We didn't find anything in there, but the deleterious variants that we were most interested in actually appear to work in the same networks as known hearing loss genes, and this obviously makes sense in terms of the logic behind how we thought about this. And so we think these are our top candidates for these genes, although not much is known about these 6 potential candidate genes either. And then I wanted to share one more interesting result with you here, and that is what we did is we took, I mentioned there were 78 known genes for hearing loss at the time. So we looked in the set of 78 genes, and for each of our individual probands, so these are labeled 1 to 13 on the x-axis, and then each of our families, family 1 is F1, family 2, and family 3, and the purple bars just indicate the parents who are unaffected. We went in and we counted essentially how many total variants we found in known hearing loss genes. And it turns out that this looks fairly the same, very even between the probands and the families. We see something like, between 15 and 20, sometimes as much as 26 variants in known hearing loss genes in each of these individuals. We then looked at how many of these variants of the exact same variant had actually been associated with hearing loss, and we see that in many of these cases there are multiple variants that had been associated with hearing loss before that are found in these cases. And then finally, we counted the number of genes that contained variants, and we see some, on average between 10 and 15 genes in each of these different individuals that contain 10 to 15 hearing loss genes that contain variants. So this was surprising, and this also gave us a little bit of insight into this process of how hearing is actually diagnosed. So when you go in for a hearing loss screening, a genetic screening test for hearing loss, you're checked for mutations in a very small set of genes, and and we're seeing that there might actually be multiple mutations in many of these genes, and so this probably isn't the best or definitive proof that that gene actually is what's causing the mutation. It turns out when we go and look in each of these individuals, most of these variants are not actually going to be causative, because most of these are in a heterozygous state, and that variant had only been shown to affect hearing loss in a homozygous state in the literature. So many of these actually don't explain the hearing loss in these patients. But nevertheless, we were able to make some headway in the 13 probands. So in five of these 13 probands, we found likely causative mutations. So these were rare homozygous predicted deleterious mutations in known hearing loss genes. So these are probably our top candidates. And in two of these probands, we actually found two separate genes that are equally likely, in our opinion, to be causing hearing loss in these patients. And at this point, we can't really say much more until functional studies are done to see which of these is actually the causative variant. In the remaining 8 probands, we didn't find, known homozygous predicted deleterious variants in hearing loss genes. What we did find, and we were able to do this because Ingenuity variant analysis is actually integrated with Ingenuity pathway analysis. So what we were able to do is we were able to find homozygous predicted deleterious variants in a gene that wasn't a known hearing loss gene, but that was one node away in some biological pathway from a known hearing loss gene. And these pathways were all curated by Ingenuity and housed in their pathway analysis software. And I guess none of these variants were found in the unaffected family members from our cohort, so that made us a bit more confident that these might actually be disease causing. And so in the end, what we were able to do is from our various examples of different types of hearing loss and different familial situations, is we were able to find in one family a novel candidate gene, in another family 2 potential candidate genes, third family 6. I don't know why that says 9. Sorry about that. And in most of our probands, we were able to find highly likely causative genes. What's really nice about the fact that we used Ingenuity is that now that this is published, anyone can actually go to this website, this variant analysis website on Ingenuity, and this is our data, and you can go here and you can kind of play around with these filters, change them as you wish, and see the data and decide how you want to actually try to filter these in different ways, and see if there are interesting hypotheses that you come up with in terms of what might actually be causing these variants, causing the hearing loss in these cases. So. I mentioned that the other thing we did, and I'm not going to spend too much time talking about this, but the other thing that we did was we also took an association type approach, and we were also looking for copy number variation. And we did in fact find we did a CNV association study genome-wide, and we actually found one novel locus that showed a deletion in 40 of our cases and only 15 of our controls, which gave a very high odds ratio. And we think that this is actually potentially a novel hearing loss locus. And in addition to this, we also found CNVs in many known hearing loss genes that seem to be underlying the hearing loss in individual cases from our cohort. Okay, so I explained to you a bit about how we use these two different ideas about the genetics underlying human disease to look at a complex phenotype like hearing loss, and we showed that both both paradigms apply in that situation, and that in some cases we do have a large subset of the population having the same variant, having the same cause, and in other cases we have each individual actually having their own private genetic cause. So now I'm going to turn to the special case of very rare diseases, and it turns out that rare diseases are quite common. So a rare disease is considered, or a disease is considered rare when in the US it affects fewer than 200,000 people, and in Europe when it affects fewer than 2,000 people. And to date there are about 7,000 known rare diseases. Most of these occur in children, and about 1 in 10 Americans is living with a rare disease. So that's about 30 million Americans. And actually 350 million people worldwide are affected by a rare disease. So that's what I mean by rare is common. Each individual disease occurs in very, very few individuals, sometimes only a single individual. But collectively, rare diseases are quite common. So I'm going to tell you a story about the discovery of a new rare disease that I was involved in. And through the story, I'm going to demonstrate some of the challenges we faced along the way. And then I'm going to spend some time at the end just discussing how our work at the Rare Genomics Institute is trying to address some of these challenges. So during my postdoc, I was working at Mike Snyder's lab, and I was handed a case. We had an individual patient, and we had genome sequencing data from the whole trio. And this patient had a very rare disease, and the family was sort of at this point where they didn't know what to do anymore. So they went to top researchers around the country. They had us and a group at Harvard and a group at Baylor all working on their data, trying to figure out what can be causing their child's condition. And so I came into this after another postdoc had actually worked on it as well, and I was handed a bunch of sequencing data from Illumina and Complete Genomics. The variants had already been called using a pipeline called HugeSeq, and then I called them also using real-time genomics, which had just come about at that time. And then we also got variant calls from Complete Genomics. So we had SNP and indel calls, and we also had structural variation calls. And I just highlighted here, we use two different platforms. So we use HugeSeq and RTG to do the variant calling. And one of the first strategies we employed was to look for variants that were concordant from both different platforms. And this is something that's done very often in genomics, where in order to narrow down your hypotheses, to narrow down your list of potential candidates, you actually take the intersection of the two sets of calls from different algorithms. And so that was something that was very standard in the field, so we decided to do that. And this was our strategy. We had the sequence data, we called the variants, and then we tried to filter and figure out what and identify what the most significant variants can be. And so when I attempted to do this, I used Ingenuity, and I followed a very similar filtering scheme as I had done previously. And I was told that the patient had a severe liver phenotype. So I did all my filtering, whatever, and then I came out to this point. There was something like 6,000 predicted deleterious variants. So then how do I now go and apply a biological filter when the patient's phenotype is so broad, and it turns out I didn't even have all the information when I was doing this And so I was told that the patient had a liver phenotype, and so I found this one gene that was transmitted in a recessive way. It was called by both huge-seq and real-time genomics. Compound heterozygous mutations in this gene. And it turns out that this gene is known to bind to another gene, and mutations in this other gene were shown to cause liver disease. So I thought, okay, this is it. This has to be what's causing this phenotype. And it turns out that I was wrong. And the reason that, one of the reasons I was wrong is because I used this idea of concordance between huge-seq and real-time genomics variant calls, and it turns out that real-time genomics, or the actual variant didn't pass the quality filter in the real-time genomics calls. So as I was doing this, I took this back to the physician who was working on the case from Stanford, and we discussed and whatnot. And it turns out that the real reason this case got solved, and in the end we were able to define a new disease, is the physician came back to me and said, well, actually, we just heard of this person at Duke who was actually collecting a very, very small cohort of patients with very similar phenotypes. And this connection is actually driven by the parents of the families. And it turns out that in the end, we were able to collect 8 different cases, 2 of which, 2 different sets of siblings, along with 4 individual sporadic cases. Our patient here was patient number 3. And by having this connection made, we were actually able to use that, and use the genomic information from all of these patients to then narrow it down and figure out that the causative gene is actually this NGLY1 gene, which did come up in my analysis previously, but there was no real reason for me to choose this over some other gene. And so really the point that I wanted to make here was that the connection between finding these second cases, second, third, fourth, and now 8 cases, is actually what led to the solving of this case and being able to define this new syndrome. So all I'm showing here, this is from the paper where we reported that so we had 8 patients in total in the end, they all share 3 phenotypes, which is developmental delay. They all had some kind of movement disorder, and they all had hypertonia. And then many of them shared, as you look across the x-axis, a decreasing number of them shared other phenotypes as well. And so I used this in my personal experience with this case, which, it kind of went on for months, and we went back and forth a lot trying to decide what strategy we should use to filter and think about these things. But in the end, no way that we filtered it was sufficient until we were able to find a second case. And so this brings me to talk about some of the work that I've been doing with the Rare Genomics Institute. So the Rare Genomics Institute is a nonprofit organization that was formed about 5 years ago, and its main purpose is to connect patients with rare diseases to genomic solutions. And we do that by essentially harnessing the connective power of the internet. So the first incarnation of the Rare Genomics Institute, when it was founded, our founder is Jimmy Lin. The first incarnation was to give patients access to genomic sequencing. So many patients come to us and they've undergone this long diagnostic odyssey. They've been through a barrage of testing. They have no idea what's wrong with their child, and they come to us both financially depleted and emotionally depleted because they're unable to figure out what's going on. And so we have we set up partnerships with academic institutions and private institutions around the country, which all provide CLIA certified whole exome sequencing. And so we're able to connect our patients with these sites around the country, and they can go to somewhere that's geographically convenient and have their sequencing done. And that was the first big push that RGI really tried to put out. And we were able to help a few hundred patients kind of begin this process. Many of them have actually completed this process. And now Rare Genomics has actually set up, or we are in the process of setting up sites all around the world. So we've established sites in the UK, China, India, Israel, Singapore, Korea. Many of these are now getting on board. And these are actually going to run as separate institutions, but all kind of connected under the Rare Genomics umbrella. So that was a huge effort to bring access to these patients, to bring genomic sequence access to these patients. And now, so I lead the research endeavors of Rare Genomics, and our team was essentially established to meet a need that turned up after this whole sequencing had been done. By the way, I should mention that in order to get their genome sequenced, many of our patients do come to us financially depleted, and so one of the things that we do is we help set up crowdfunding platforms for them so that they can raise funds to get their genome sequenced, because insurance, in most cases, did not cover the cost of sequencing. And so that was one of the first way in which we used the internet to drive research forward. So the research efforts now are here to kind of fill this need that we saw appear, in that many patients who go to get their genome sequenced at many of these academic sites come back with very unsatisfactory results, and in most cases, no genetic diagnosis. And in sort of a standard genetic sequencing operation, there really isn't a whole lot of in-depth analysis that goes on the other on the part of the sequencing facility in order to figure out what's causing this disease. And for the case of rare diseases, it's even, as I just kind of demonstrated with that previous story, there really is a need for very in-depth analysis. There were many different researchers working on the NGL1 case, and we weren't able to come up with anything until we found the second case. And so we really do require a lot of in-depth analysis. So my team is trying to build and develop a model of research that we think is going to address many of these issues that come up with rare disease genetics. So the process that we're using is this. In the first case, and I mentioned that in my experience, I wasn't completely informed of every single phenotype that the patient had. So the first thing that we're trying to do is compile very well-defined and complete medical phenotypes, medical histories, collections of symptoms of each patient. And we're doing this via a patient-driven cloud platform called Patients Knows Best. So the really nice thing about Patients Knows Best is that each patient has their own account, and they put all of their data in this one place so that it's accessible by anyone who's given access. But the patient is the one who controls the sharing, and the patient is the one who controls who they give access to. And so that way we overcome a lot of consent issues, and we overcome a lot of logistical issues in terms of handling data and having researchers share data, because all the data is in this one place and you just need to be given access to it. I work with a team of 20 very amazing volunteers, most of whom have PhDs in some biological field. And these people do amazing work in that we go and we do very in-depth genetic analysis and reanalysis that leverages the medical history and leverages this well-defined, complete set of medical phenotypes that we've compiled. So we perform this very in-depth genetic analysis, which often is not done at a standard sequencing process. We also extensively compile literature that relates to the symptoms or the genes of interest or treatments or similar cases that exist out there. And often, I should say, similar cases are not reported in the literature, but many times for rare diseases, similar cases are reported somewhere on the internet, whether it be a blog or a Facebook page or someone's private page about their child. And so we do a very kind of complete search to try to provide leads for our families. The other thing that we do, so what I'm saying here, some of these features are provided by the physician or the genetic counselor, but in most cases this isn't really their job, and so this is where we fit in to fill that need. The other thing we do is we provide scientific recommendations based on the unique situation of each case that we think would help drive research forward. And as I mentioned, most of our teams are actually researchers as their day job. So these are people who know what they're doing. Finally, we connect patients to relevant experts who we know may have been, who we know have been working on a particular gene or working on a particular treatment or syndrome. We also connect patients to relevant clinical trials. The two really, I would say, different things that we are doing in the research model are the last two. And the first is that we're really trying to establish a global collaboration in order to solve these cases. And we do this through crowdsourced research. And what I mean by that is each member of my team, each analyst on my team, when they work up a case and they do all of these things I just described, we actually end up writing this very in-depth scientific report. And we post these reports. Of course, these aren't going to be fleshed out like a publication. We don't do molecular studies to follow up and all of that. But we do have a large amount of research that's communicated to the world in these research reports, which we post on our online forum. And so this online forum actually is a place where anyone who's interested in working on any of these cases, any researchers, anyone in the community who would like to look at what's been known about this case and what more they can do, can go here and you can give your input. You can volunteer experiments to be done in your lab. You can interact. You can ask questions. You can ask for further analyses to be done, and so on. So anything that can help drive this research forward, and as we've seen in many times, global collaboration is really the reason that many of these rare disease cases get solved, because by definition there are so few of them that you really have to search the whole world to find a second case, not just your country, not just your city. And then the last thing that we're trying to do is build an intelligent database that curates genotype, phenotype, treatment and outcomes of treatments. And this is something that we think is going to be really, really important in helping to connect and for patients to find that second case that's really going to be critical in helping to figure out the genetics behind the disease and help provide a diagnosis. And so this is the model that we've been working with. And really briefly, I just want to tell you, so we've had a pilot program going on now for about a year, and so far we've completed 9 cases, which are all on our forum, and then we our forum URL is up here. So if you're interested, you can go and take a look at some of these cases, and maybe some of you have some input for us. But so far we've done 9 cases, and we have about similar number in the pipeline that are currently being worked on. But for about half of these cases, we've come up with some pretty sound findings. So many of them we found gene, we found in this case we actually were able to define the syndrome, and the patient got referred to an outside expert. And this came about because of some back and forth between us and the patient's physician. This one was actually referred to experts for a trial treatment. In this case, the patient's family actually found the second patient. The patient is American, but the second patient was found in Australia via some internet communication. And we're hoping to get that second patient through our process as well in order to help both of them. And in the other cases, we've managed to narrow it down to a very short list of potential genetic candidates, but also provide a lot more biological insight into the case for the family and for the physician to work on. And so we think this model is actually going to be quite successful in helping some of these families figure out, what's going on, and we're really excited to apply it to more cases. And I should say, Rare Genomics Institute is entirely volunteer-based, so we're always looking for more volunteers. So if any of you in the audience are interested in taking your expertise and applying it in a nonprofit sector, we would be very, very happy to help you. And we have roles with varying amounts of commitment, so almost anything is possible in terms of involvement. So with that, I just want to recap what I spoke to you about today. So we discussed 2 different paradigms for understanding the genetics underlying human disorders. From this, we were able to use the example of hearing loss to extract probably 3 important aspects of identifying mutations with hearing loss, which is that exome sequencing of families can reveal novel mutations, but not in every instant. Sequencing a small number of isolated probands can actually also lead to some candidates, but again, not in every case. Some cases remain quite challenging. And finally, CNV analysis is also important in hearing loss, and as we've shown, we were able to find several loci associated with hearing loss through doing CNV analysis, and shown that this is a type of variation that should be investigated. But more generally, we see that it's important to employ both familial and association strategies to successfully identify rare and potentially private mutations, as well as more frequent mutations that might be underlying some complex human disease. And finally, we've shown that exome sequencing, coupled with very deep analysis that leverages the medical history, global collaboration, and essentially, the power of the internet to connect people and research and data, can actually be very, very insightful for discovering the underlying genetics of rare disease. So with that, I would like to just thank everyone who's contributed to some of the work that I've talked about today. Everyone on the left has contributed to the hearing loss study. This work was done in Mike Snyder's lab in collaboration with Ira Shriver, both at Stanford, and the exome experiments were done by Fareshta, a postdoc in Mike's lab. The NGLY1 deficiency work, these were my collaborators at Stanford, and then, of course, there are many other collaborators at other institutions who all contributed to the final process of defining this new syndrome. And finally, I would like to thank all of the volunteers on my team at the Rare Genomics Institute, as I said, these people give a lot of their time to do very in-depth, high-quality use cases, and I think it's really paying off. So with that, I would be happy to take any questions.